Thank you very much, Gordon, and I am deeply honored to be given this prize, and I'm very grateful to the Microbiology Society, not only for the award, but for putting a spotlight, a literal spotlight, on a clinician scientist. We are a dwindling community with like a 6% decrease in numbers over the last decade and a quarter loss in numbers of senior lecturers. I often not sure if I consider myself a proper scientist, but I hope by the end of the talk you'll be convinced that we are of some value in bridging the kind of gap between the bench and also the bedside. So I'm going to start, I'm going to take you on a journey between Scotland and Malawi, and I'm kind of representing in Scottish colours and a bit of African colours today, just to kind of represent that. I'm an infectious disease clinician. My passion for infectious disease really developed when I did my medical elective in 1999 in Malawi in a large teaching hospital called Lilongwe Central Hospital. So during that time, it was really the height of the HIV epidemic, where there was no available antiretroviral treatment, and really left a huge impression on me and really led me down a path to train in infectious diseases. And it turns out I undertook my infectious disease clinical training in Glasgow, and it turns out it's a very exciting place to train in infectious disease. In the time that I did my training, I dealt with outbreaks in E. coli 0157, Clostridium novii, anthrax, also managed cases with Crimean Congo hemorrhagic fever and Ebola. But actually one of the most exciting times, and really kind of led me to develop my passion for influenza, was when I was on call at the University Hospital Monklands, which is a district general just outside Glasgow, where I treated the first 2 UK cases of influenza A H1N1 in April 2009. And really kind of as a clinician, watching this new virus unfold was really interesting, because the patients that I was admitting during that time looked very different from the normal patients I would admit with flu, which were elderly with lots of underlying health conditions. But in fact, I was admitting patients much younger, often overweight, but also those that were kind of peripartum as well, which was very, very unusual. And this made me think maybe perhaps I wanted to pursue a career in clinical epidemiology, really kind of trying to take a step back and understand pattern and frequency of diseases. So how does a clinician try to become a researcher? It turns out it's pretty challenging when you don't work in an academic unit. So I naively applied for a number of clinical PhD programs, of course were unsuccessful. And after seeking advice from a senior academic, he basically told me my CV was rubbish, which was fair. But I took his advice, and he's like, if you want to become a clinical epidemiologist, you need to take time out and perhaps do a master's in epidemiology. So that's exactly what I did. I basically, in the penultimate year of my training, so I was about 16 months from completing my infectious disease training, I decided to do a full-time master's at the London School of Hygiene and Tropical Medicine. And on my 5th attempt at applying for a PhD program, I was successful. So I got one through the University of Liverpool, and basically spent the next 3 years in Malawi doing my PhD fellowship. So I combined basically my passion for flu in Malawi and decided to do my PhD fellowship looking at the impact of HIV on the burden and severity of influenza illness in Malawian adults. And at that time, flu had never really been studied. CDC had just implemented a respiratory viral surveillance program the previous year. I was based in Blantyre, which is where the Malawi-Liverpool Wellcome Trust Clinical Research Program was based, and there was my research. I did 2 prospective observational studies. The first study involved in recruiting a cohort of 600 HIV positive and negative individuals. I followed them up over 2 years. They basically came for regular visits, and we also educated them on what flu-like illness was, and they came to the clinic and saw my study clinician when they had flu-like illness, and we basically took swabs and detailed clinical history from them. I also did a case control study where a second research team basically were based at the emergency department at the large Queen Elizabeth Central Hospital and the adjacent primary care clinic, where they recruited patients with severe hospitalized pneumonia and also those with mild influenza-like illness. And essentially, we tested all of these recruits for influenza and basically compared those with mild and severe influenza to look at risk factors for severe influenza. So in brief, my PhD fellowship found that those who were HIV infected had 3 times increased risk of symptomatic influenza and 5 times increased risk of severe influenza, with additional risk factors being household crowding and food insecurity for symptomatic influenza and poor sanitation, and also food insecurity for severe influenza. And really that highlighted to me for the first time that risk factors for a common condition may differ dramatically depending on the setting and kind of underlying epidemiology. In the community-acquired pneumonia cohort, we also found that respiratory viruses were very prevalent, with 1 or more respiratory viruses detected in over a third of patients. About 9% of them were influenza. And of note at this time, influenza vaccination was not available in Malawi and still isn't routinely available publicly, although it can be acquired by private prescription. And also the kind of etiology that are identified also differed substantially to what I'm used to in the UK and other high-income settings, with tuberculosis featuring in just under a quarter of patients, and also other etiologies such as PCP, which kind of tells you that HIV really drives pneumonia etiology in the setting, particularly in Malawi, where HIV prevalence at the time was in the top 10 in the world, was around 10.6% nationally. So I also, at that time, also ran the hospital-based respiratory viral surveillance that was funded by the CDC because it was recruiting in parallel to my study. And essentially, we identified kind of annual cycles of influenza, but not really any clear seasonality. So it's not clear that if influenza vaccine became available, when would you kind of deploy it? It's not like there's clear winter seasonality like we see in the UK and other temperate settings. So when I completed my PhD, I returned to complete my clinical training. And in Scotland, we don't have NIHR clinical lectureships, which basically give you up to 50% of research time, really to kind of continue your research work. I applied for one of these posts in Liverpool and was unsuccessful, but fortuitously I had asked Professor Emma Thompson, who is now our director at the CVR, for a mock interview, and she said to me, like, if you are unsuccessful in this, come and talk to us. And lo and behold, a year later, I find myself being appointed a clinical senior lecturer at the MRC-University of Glasgow Centre for Virus Research. So, Gordon's already given us an introduction of the CVR, but for those of you unfamiliar, it's based in Glasgow in this lovely gold building. I've just put the 2 hospitals. I also do 20% clinical work at 2 different hospitals, but the CVR is a large collective of researchers. I don't know what the collective name for virologists is. 8 years later. But basically, as a large collective of researchers that study human and animal viruses that impact on human diseases. And really, we span everything from kind of molecules to population. We heard David Bella being mentioned in Alan Parker's prize lecture this morning, who does a lot of work on like cryo-EM and structural virology. And we also have kind of real expertise in genomics and bioinformatics, field epidemiology, arbovirology, towards the kind of more clinical and population-based kind of virology, which is where my work had traditionally sat. When I joined, I mean, I honestly, I kind of genuinely felt like a bit alien myself because, I'm a clinician, and there are apart from Emma, there are no other clinicians. And really, I had to spend a lot of time to really kind of, in terms of just moving institution, really to kind of understand fundamental virology and really understand the work that all my colleagues do and how I can possibly integrate this into my work. But all my colleagues were really extremely welcoming, and really the pandemic then came about and really accelerated that integration very rapidly. So in the kind of initial 12 to 18 months, I was really kind of trying to find my feet. So also kind of thinking of how I can pivot my work from more African focus to UK focus. I also had a number of interactions that proved to be quite important later on. So I presented some of my Malawi flu work at an ECDC meeting in Stockholm, and I met Dr. Gail Carson, who's the Director of Network Development for ISARIC, which is the International Severe Acute Respiratory Infection and Emerging Pathogen Consortium, a real mouthful, but a really important organization that I became very involved with, that basically set up a standing protocol that gets activated for any emerging infection. So I thought, oh, perhaps the CVR should be part of this. And she introduced me to the two UK chief investigators, Kenny Bailey and Callum Semple. And in September 2019, I basically led the CVR to join this consortium. I was also asked to present influenza burden data from Malawi at a WHO meeting in Geneva, and there I met the head of the WHO African region for the flu program, and she had just advised me that Malawi had just set up their own public health institute, which wasn't there when I was doing my PhD. So a few months later, I visited Malawi and essentially met the respiratory viral surveillance team from the public health and basically helped them design the National Respiratory Viral Surveillance Program. And I also met the director for the Malawi Epidemiology and Intervention Research Unit, which does a lot of kind of population cohort work, chiefly focused on previously it focused on HIV and TB, but now with increasing focus in non-communicable diseases. And again, that becomes important in just a few months' time. I also was thinking of how I can pivot my work to the UK, and working as a clinician, I realized there was a real gap in looking at risk stratification tools for hospitalized influenza. So in kind of routine clinical practice, we use a lot of risk stratification tools. The kind of news score that is basically, this chart is present in every single patient that is admitted to hospital, where the nursing staff records routine physiological parameters daily, and basically it comes with a score, and if it increases to a certain level, you basically highlight to nurses that the patient is acutely deteriorating and may need kind of medical input very rapidly. There's also the CURB-65 score, which is used in community-acquired pneumonia, and again, really helps clinicians, particularly more junior members, trainees, to kind of decide on whether someone needs escalated or maybe they're safe enough to be discharged. So given that every winter we are inundated, including the winter that we just had there, that there are many patients being admitted with influenza, I realize there isn't a risk stratification score for influenza. And looking at kind of existing scores used for pneumonia and sepsis, they didn't really perform well. So I did a retrospective cohort study, included patients admitted over a 3-year period across all Greater Glasgow and Clyde, patients, over 1,000 patients, and developed this kind of 5-parameter influenza severity score. And again, as someone who, until very recently, was often admitting patients at 3 in the morning, that any score has to be very easy to use, that doesn't require a huge amount of mental arithmetic, but also have parameters that are readily available and not involving kind of complex investigations, which is why it's kind of very important that the scores just contain very simple metrics. And we found that I developed a score that with increasing score was associated with increasing risk of mortality, and it performed much better than all the other existing risk scores. And of course, this score, I had written up the manuscript, ready to be submitted in January 2020, when the pandemic occurred, and therefore rendered this manuscript completely obsolete, because there was never a reality where flu was going to exist without SARS-CoV-2. So anyhow, the lesson is that no effort is wasted, as I will explain later on. So, 2020, the pandemic happened, and it was a real kind of baptism of fire for an early career researcher. You know, clinically, when I was doing clinical work, the infectious disease unit was converted to a COVID high dependency, and I also got kind of thrown into the deep end and really got really afforded a lot of opportunities in terms of participating in government advisory panels, doing a lot of media engagement. I will also explain later why I'm dressed like a police person there later on. But I'm not really going to talk about the kind of advisory and public engagement work today, and I will kind of focus on these key 4 aspects in the next few slides. So very early in the pandemic, when we knew absolutely nothing about SARS-CoV-2, we decided to turn our attention to what the kind of existing human endemic coronaviruses, of which there are 4, OC43, NL63, 229E, HKU1, and using kind of 13 years of primary and secondary care testing data across Scotland with a postdoc that was based at the CVR at the time, Seema Nickbash, we basically identified very clear winter seasonality for all 3 of these coronaviruses. And we also analysed the respiratory viral surveillance data in Malawi, the hospital-based surveillance data over a 7-year period with a PhD student, Dori Kovacs. And essentially, we found very different patterns, with most of the seasonal coronaviruses showing biannual peaks, which again demonstrates that seasonality may differ in kind of tropical and subtropical regions. We also kind of published one of the kind of landmark systematic review and meta-analysis summarizing kind of key early studies that looked at SARS-CoV-2 viral load shedding, viral load dynamics, as well as infectiousness. Of course, we now know all about this, but in the kind of early months, there were lots of studies coming out deploying very different methods of, detection, different patient populations. So summarizing this was a bit of a challenge, but we basically identified different shedding durations by site, that viable virus were rarely detected beyond 9 days, and that PCR positivity didn't equate infectiousness. And peak viral load occurred very much around symptom onset, which might explain the more efficient transmission relative to SARS-CoV-1 and MERS-CoV, and highlighted the importance of rapid case identification and early isolation. So this systematic review really informed kind of modeling as well as isolation policy in the first year of the pandemic. As I said, we became kind of really, the ISRIC study became like really critical to our SARS-CoV-2 research work. So I led the recruitment for the study, so we activated for COVID-19 and became one of the largest prospective cohort studies recruiting hospitalized COVID patients. So I recruited from 16 research sites across Scotland, and the CVR, because it has excellent CL3 facilities, became the outbreak lab for Scotland. So we received in excess of 10,000 samples of varying types from all the sample sites to the CVR, and then distributed them to a number of labs also. And over the 2 years where we recruited, we collected data from over 300,000 patients across the UK, and also obtained biological samples for just under 3,000 patients, of which a quarter of them were from Scotland. And because of this huge numbers, really, this study really advanced our understanding not only in how COVID-19 manifested in patients, but also kind of host characteristics and virus characteristics of the study as well. And just to highlight that, lots of other CVR and University of Glasgow PIs and researchers really led large pieces of important work in various aspects using the ISRIC infrastructure. So for myself, apart from recruitment lead, I was also in the clinical characterization work package, and I led on risk stratification. I told you no work is wasted, so I basically, having spent a lot of time thinking about risk stratification, it was obvious in the early days of the pandemic that we needed a new risk stratification tool because COVID-19 was really overwhelming health systems worldwide, and the clinical course was really very different from that of influenza and pneumonia. And so effective triage of patients for clinicians at the front door to understand who can be safely discharged or who needs early escalation is critical. And a systematic review published in the BMJ quite early on in the pandemic had identified a large number of existing prognostic tools that had been developed, but a lot of them had high risk of bias based on single center data and had small sample sizes. So we wanted to develop a pragmatic clinically relevant tool to help clinicians at the front door to predict in-hospital mortality. So I'm not going to go into the specifics of this, but we basically took candidate variables from the literature, and because we need it to be applicable to most people, we excluded variables that were missing in more than a third of people, and we used the traditional modeling approach as well as a machine learning comparison, and basically developed it using a derivation data set and using a later data set involving altogether just under 60,000 patients to derive and also internally validate our score. So we developed a 21-point score. That comprises basically some basic patient characteristics, admission physiological parameters, and some easily available blood tests on pretty much all admissions. Just to highlight how important age is that age comprises 7 out of the 21 score, and to help clinicians interpret the score, we divided this to kind of low, intermediate, high, and very high risk, which corresponded to mortality of 1, 10, and over 30% or more. So in reality, how was this used? Basically, if a clinician, based on clinical judgment, felt that a patient was potentially well enough to be sent home, if they had a low risk score, then it gives them more confidence that this was appropriate, whereas perhaps a patient that scored 4 or more maybe required earlier escalation to critical care if that was deemed appropriate. So of course the score was developed using kind of very early data. Most of the patients were infected with the ancestral virus, and none of the patients had been vaccinated. We wanted to understand if the score continues to accurately predict in-hospital mortality in vaccinated individuals infected with different variants. And studies around the world have shown that remains true, and to date the study has been externally validated in more than 200 studies from 30 countries, and have been included in both UK and international policy guidelines. So I also was involved in kind of characterizing kind of bacterial co-infection in hospitalized COVID-19 patients, because this was really not well understood. And remembering early in the pandemic, there was really very little treatment options available to clinicians, with the exception of supportive treatment such as oxygen therapy and kind of inotropic support. And so a very high proportion of patients received antibiotics, because also a number of the characteristics that clinicians use to decide whether to give someone antibiotics, such as having raised C-reactive protein or having chest X-ray changes, they were often frequently present in patients with COVID-19. So unsurprisingly, in the first wave, we found that 88% of patients received empirical antibiotics, when in fact less than 2% of patients had community-acquired bacterial co-infection. And actually also the pathogens that were identified were dissimilar to that frequently identified with influenza, where co-infection rates of up to 23% are described. So we rapidly kind of released this information and made sure that we didn't, blame clinicians for giving too much antibiotics, but it's clearly very important for antimicrobial stewardship. Particularly, we just had a session on AMR here, when vast majority of patients being admitted at that time were COVID-19 patients, and that a lot of the empirical antibiotic was really unnecessary. We have subsequently done some work with UK Health Security Agency across all 3 waves and continue to show that basically infection there's very little kind of bacterial co-infection detected on admission, and actually that proportion is only marginally higher in those that have been hospitalized for more than 48 hours. So changing gears a little bit, at the same time I was also asked by Public Health Scotland to help them develop a syndromic surveillance for severe acute respiratory illness. So this was recommended by the WHO in 2021, and prior to the pandemic, SARI surveillance is mostly restricted to kind of flu test positive cases, and they wanted to make sure that they capture proportion positive for flu and COVID based on a kind of clinical syndrome. So we developed this in the Queen Elizabeth University Hospital in Glasgow, which is one of the largest hospitals in the UK with over 1,100 beds. And we developed this, the Charisma study, really to kind of capture the impact of SARS-CoV-2 and influenza hospital admissions in Scotland in real time to kind of describe the evolving epidemiology, kind of risk factors for severe disease, and also characterizing kind of viral, and viral bacterial co-infection. And in just over 4 years, we've recruited just over 2,000 patients. So we have a research team that sits in the emergency department that screens eligible patients for severe acute respiratory infection. If they are eligible and consent to be recruited, we obtain an upper respiratory tract swab that undergoes the routine kind of flu and COVID point of care testing that is recorded in an electronic case report form. And then we store the sample for kind of additional research diagnostics. So we do multiplex PCR, and we are doing metagenomic sequencing on these. And we've also used this platform to evaluate kind of novel point of care diagnostic tools, and also recruited patients into commercial studies evaluating novel treatment, most recently an IL-33 inhibitor for respiratory viral infection. But essentially, the kind of data that's captured on the ECRF goes into the trusted research environment and is combined with routinely collected healthcare data. So Scotland has a unique identifier for every resident that basically allows very easy matching of their health records. So we just basically combine the research records with routine health records, and this data is provided to Public Health Scotland weekly, so they're just really a week behind on kind of percentage positivity for flu and COVID that goes into their weekly report that's issued during the winter months. This is just really to show you what goes to PHS, that the number of cases positive largely reflects the kind of national numbers of COVID-19 and influenza admissions in the country. And some of the kind of key findings that we've identified, so this is some data from year 2 and 3 recruitment, is that we do kind of multiplex PCR at the CVR, and this is work done by Ellen Vink, one of my PhD students. Firstly, we identified that multiplex PCR identified one or more respiratory virus in nearly 46.5% of patients, compared to just 20% using the point of care tests, which is flu and COVID, which is basically what most patients routinely get. But also, if you have a positive flu or COVID point of care, you are much less likely to receive empirical antibiotics, 68% compared to 90%. Additionally, we find that chronic lung disease, patients with chronic lung disease, which comprises over 50% of the cohort, is an independent predictor for infection of non-flu and non-COVID viruses, because a lot of them present with viral exacerbations of chronic lung disease. So there's definitely a kind of opportunity for antimicrobial stewardship to really reduce unnecessary antimicrobial use by identifying kind of more respiratory viruses early on. So we're planning to deploy a kind of rapid multiplex point of care test to, particularly in this chronic lung disease group, and so watch this space. So I'm taking you back to Malawi now. So in parallel, we also did a lot of research work in Malawi during the pandemic. And particularly in the early months of COVID-19, there was a real concern that there would be higher morbidity and mortality in a lot of African settings because of a high prevalence of underlying comorbidities such as TB, HIV, and malnutrition, but also kind of an absence, lack of health infrastructure such as critical care facilities, as well as just even supplemental oxygen. So, led by priorities identified by the Ministry of Health, which is looking at whether healthcare workers were a risk factor, but also kind of building kind of serological diagnostic capacity, we developed a study, a prospective cohort study, which was small but really rapidly deployed using some GCRF funding, where we did a prospective cohort study of healthcare workers and community, healthy members in the community, and over kind of June, July, August. And during this time, the Ministry of Health policy for testing was really by epidemiological risk, so people who had traveled or been in contact with someone who'd been unwell. But what our study identified was a really high prevalence of active infection and rising seroprevalence in a largely asymptomatic cohort. So very few patients, very few participants developed any symptoms at all. And we used an in-house ELISA developed by my CVR colleague, Brian Willett, who's a viroimmunologist. And this study was run by two researchers at the University of Health Sciences, Tony Nirenda and David Chima, who also supervised this serology work in Blantyre. And this really generated evidence for the Ministry of Health to change their testing policy, because this kind of high proportion of asymptomatic infection was really unrecognized, and really led to highlight the importance of serological study, particularly in this setting when there's such high kind of asymptomatic and post-asymptomatic infection. So at this time, Mia Crampon, who is the director of Miru, contacted me. So she has, a large group of staff that are recruiting for population cohorts in urban Lilongwe, which is the capital city, and Karonga, which is a rural area up in the north. And like the UK, they had to pause all non-COVID-19 research. So she asked if we can develop a study to really understand the kind of population exposure and transmission of SARS-CoV-2. So we basically applied to Wellcome and got some funding to recruit 1,000 individuals in each of these sites and follow them up 3 monthly for 12 months. And we did ELISA and pseudotype neutralizing antibodies and also PCR, and also recorded a lot of clinical metadata. And for a lot of these, particularly the rural site, these are the kind of road conditions, so it's not possible to get a vehicle down these roads, so a lot of them use kind of motorbikes. And so the reason I'm dressed as a policeman is because when I went to visit, they insisted that I wore the spanking new tabard and also brand new welly boots in 40-degree heat for 7 hours, which was stressful. But it was amazing to see what the team did. We also taught the participants to do self-swab, because in Malawi, Ministry of Health kind of PPE requirements required kind of full-on hazmat suits, and we couldn't afford to do that for 2,000 participants at 4 time points. And actually, this went down remarkably well. So what did we find? We basically found increasing seroprevalence over time, always much higher in the urban area compared to the rural area, but also had differential seroprevalence by age, with yellow being the younger age group, under 15, and the other 2 lines are the adult age groups. But as you can see, over time, basically, the children catch up and the rural area also catch up. So by the end of the BA1-2 wave, essentially over 90% of both populations were seropositive, despite that less than 20% of them had received vaccine. And actually, by survey 4, the seroprevalence did not differ by vaccine status. So again, this was fed to the Ministry of Health very rapidly, and at that time they were vaccinating everybody, and they had then switched to vaccinate target groups. But what target groups should be vaccinated? So we also did pseudotype neutralizing assays developed by, again, Brian Willett, and we had a very talented PhD student called Barry McCormick, who did, I think, about 4,000 neutralizing assays, fixed dilution, and also titrations of the positive samples, and we identified essentially that those aged under the age of 15 had the lowest neutralizing antibody response with infection. For all variants, we found that those with hybrid immunity, which is those that are infected and vaccinated, had the highest neutralizing antibody response compared to those infected or vaccinated alone, which is concordant with what we find in other settings. And that HIV-infected individuals, particularly amongst vaccinated individuals, were generated much lower neutralizing antibodies. So perhaps particularly, definitely HIV-infected individuals should be part of the targeted vaccinated group, and we weren't able to look at other risk groups due to the kind of low prevalence of other comorbidities within our cohort. We also found, because we were able to look at different variants in the pseudotype neutralizing antibody response, that over time, that there was an increasing complexity of immune landscape in the kind of cohort over time. We were able to capture some seroreversions, and this was particularly marked for people who had Delta infection in the first place, but that the proportion of people that were completely naive to the virus really had decreased to much smaller numbers by the fourth survey. It was a busy time during the pandemic, and I also kind of over time tried to develop the work that my group did. So I lead the clinical viral epidemiology group, and increasingly I wanted this group to integrate clinical virology, epidemiology, as well as some of the fundamental virology expertise that my CVR colleagues and also external collaborators had. And it's now I've tried to summarize them into 6 key areas. So in the last few minutes, I just will quickly go through these because I realize I'm standing between you and the pub, so I'm trying not to go over time too much. Going back to the Malawi pneumonia cohort, we've identified that 1/3 of patients, despite really extensive bacteriology, virology, and TB testing, didn't have any etiology identified. And this is a cohort with very high in-hospital, with 12% in-hospital mortality, 20% 90-day mortality. And so we wondered whether we can use metagenomic and target enrichment sequencing to identify additional viral pathogens that are not targeted by the multiplex PCR. So this work is partnered with the head of CVR Genomics, Anna Felipe, as well as Joseph Hughes, who is a senior bioinformatician, can't say the word, at the CVR, and to highlight that this is not just a problem in low-income settings. In large cohorts in the US and Switzerland, we also aren't able to identify the etiology of community-acquired pneumonia. So the CVR Genomics team have spent some time optimizing their metagenomic and target enrichment pipeline. And so we have another very talented PhD student, Kirsty Kwok, who's been carrying forward this work. So first of all, she did metagenomic sequencing on a number of the community-acquired pneumonia cohort samples from the Malawi study using metagenomic sequencing, and identified a number of additional viruses, including particularly greater diversity in HIV-positive patients. We identified rhinoviruses in 5 patients despite them being negative for a PCR, but we didn't find viruses in 64 of the 119 samples. So we wondered whether we could do target enrichment sequencing to boost the viral signal. So we decided to deploy the Twist Bioscience as a commercial pan-viral panel and also a respiratory panel. But we first of all checked whether it would have good coverage. So we downloaded all the respiratory virus genomes available in GenBank and combined with the genomes that we already had. And it turned out that the coverage was pretty poor. And so we designed a custom secondary target enrichment panel to enhance genomic characterization. And first of all, we tested it on 11 samples and found that there was no difference in genome coverage, but much higher proportion of viral reads in the target enrichment compared to the metagenomic sequencing. And we basically did it in the 64 samples where we hadn't detected anything in the metagenomic sequencing, and identified at least one human virus in an additional 41 samples. And we are now sequencing kind of contemporaneous healthy controls, which is really important when you're doing this kind of work. We are also expanding the serosurveillance work. So we've got a Gates-funded project where this is a list of pathogens that the Ministry of Health has targeted, and we're leading work on arbovirus. So John Kelly is another PhD student who's looking at dengue, chikungunya, and Zika, and a number of serial cross-sectional serosurveys. And Dana Gabi is looking at RSV seroepidemiology in young children under the age of 5, ahead of potential introduction of RSV vaccine, perhaps in 2027 or 28 in Malawi. In terms of, speaking of RSV vaccine, so this has been introduced to pregnant women over the age over 28 weeks of gestation, as well as older adults aged over 65, and more recently care home residents. And we've got a number of studies, again using this linked Scottish data resource, which allows linkage of pregnant women and also birth and neonatal data with larger data sets to evaluate real-world maternal vaccine effectiveness. And we've also got an MRC-funded study really to look at predictors, maternal and pediatric predictors of maternal RSV vaccine effectiveness, because we assume that if you vaccinate mothers, then the infant will be protected, but we're not sure whether maternal risk factors that are prevalent in the UK, such as diabetes, obesity, and smoking, may impact on transplacental transfer. So we're just about to start this work, recruiting a prospective cohort of 600 pregnant women and infants. Last but not least, we're still responding to emerging infections. One such outbreak is the pediatric hepatitis in young children, which started it in Glasgow. And I presented this work at this meeting a couple of years ago, but in case you missed my really amazing talk, I will just briefly tell you. So a very astute pediatric gastroenterologist identified 5 young kids who basically presented with severe acute hepatitis, and that tested negative for all the usual causes, which would be viral hepatitis, drugs such as paracetamol, and autoimmune hepatitis. And so she highlighted Public Health Scotland. I basically was asked to attend the incident management meeting, which literally was 3 days after we closed recruitment for COVID-19. We activated for this pediatric hepatitis outbreak because it turned out there were a number of other cases observed in Scotland. And within a week, we'd recruited 9 patients and did metagenomic sequencing on various sample types, so blood, liver, stool, throat swab as well, and identified 2 kind of key viruses. In all children, we identified this parvovirus called adeno-associated virus 2, and in 6 out of the 9 children, we identified adenovirus. So those who have not heard of AAV2, essentially, it's a dependo parvovirus because it requires a helper virus in order to replicate. So it's pretty ubiquitous. Most of us are exposed between the ages of 3 and 5, which is the age of these children. And adenovirus is one such type of virus. And then colleagues at the CVR did kind of serology and also in situ hybridization, basically highlighting AAV2 RNA around the kind of arteries of the hepatocyte and also within the nucleus. And Vanessa Herder also did kind of codex multiplex imaging showing like an inflammatory infiltrate in the explanted liver. So a number of these children required a liver transplant compared to the control liver. And the last piece of the jigsaw was solved by the Scottish National Blood Transfusion Service, where they identified an overrepresentation of an MHC class 2 DRB 10401 allele, which was identified in 93% of the children with this unexplained hepatitis, compared to 16% of the Scottish population, suggesting that perhaps it was a case of co-infection with adenovirus and AAV2, with a massive wave of adenovirus observed in 2022 after all the kind of non-pharmacological interventions had been removed in a genetically susceptible individual that caused this presentation. And the reason I'm highlighting it is because this rapid response that we were able to generate, where we recruited the first patient within a day, sequenced the first samples within a week, and were able to kind of put what we find in a UKHSA technical briefing and release a preprint within 3.5 months, was precisely because of existing collaborations that had been established during the pandemic, and also the infrastructure, research infrastructure with ISRIC that was already in place, and all the research teams knew exactly what to do. And we published this work almost exactly a year to the first kind of identification of these. So lastly, we're also kind of expanding the Charisma study, where we're hoping to develop a multimodal decision support tool by combining the kind of really detailed clinical metadata with chest X-ray imaging, really to try and improve kind of clinical phenotyping of patients and helping clinicians decide on these patients. So I've taken way more time than I should have, so I'm going to end there. I am very passionate about training next generation of scientists. I have so many people to thank that I'm very stressed out that I would miss somebody off the list, so I decided not to include any names. But I want to thank my group, my really amazing colleagues at the CVR, my Malawi collaborators, also my collaborators at Public Health Scotland, UK Health Security Agency, lots of other collaborators across UK universities and consortia that I have collaborated with, the funders, and most importantly, the patients, participants, and the study teams that have contributed to this work. And I apologize for going a few minutes over time, and thank you so much for listening.